Hello, I'm Dr. BCMR, Assistant Professor at Shastra. We have seen, or we know, that all the organisms, apart from some propagating entities like viruses and viroids, the genetic information is made up of DNA, or simply to put, or it is made up of nucleic acids. Since the time of the emergence of those molecules on the surface of Earth, or probably somewhere else in the universe, what we now know is it has diversified into highly complex system, giving rise to various life forms or propagating entities, and it never stopped. And if we think logically or philosophically, it must have happened in a small stretch of DNA must have first polymerized, or RNA probably. And that stretch has increased in two ways, or we will say it as diversity, has increased in size, and also has increased in complexity. All the living organisms would account for these diversity, the genome size and complexity. In other ways, if you also see in terms of complexity at least, the first nucleic acid might have been just a double-stranded or single-stranded one. And say if we think about a little more complex ones, we will come to something like plasmids. Plasmids are double-stranded circular DNA molecules. If we go a little further in terms of complexity, this is not the order in which the emergence might have happened or the evolution of the organisms might have happened. But in terms of complexity, we can count the next propagating entities as viruses. Viruses have genetic information, that is, say DNA plus proteins. And upon that, if we think of complexity, we can think of simple cells. Those are bacteria and archaea, they are prokaryotes. And a more complex one would have been the animal cell, or simple protozoa. You can take of single-celled eukaryotes. Eukaryotes would have a nucleus and they would have mitochondria, and some of them would have, apart from nucleus, mitochondria, they also have chloroplast, right? And if you just think of the organismal size, we can see simple eukaryotic like these single-celled eukaryotes to multicellular organisms like the mice or mouse, any of those fish, reptiles, aves, mammals, and also humans and other primates. Similarly, with plants, we can see about angiosperms and gymnosperms. The point of writing this or depicting this here is that we know that DNA is the genetic information, or has the genetic information. The sequence of the nucleotides determine the sequence, which is the information that codes for propagation and building up of the whole body. If we see the body of the organisms, it is complex. Just let's see a cellular one, such as the prokaryotic bacteria or archaea. We can see that there is DNA, and there are a lot of chemical reactions that are happening in the cytoplasm. There is cell wall, and each of them are made up of complex other complex molecules like the carbohydrates, lipids, proteins, and of course the nucleic acids, RNA and DNA. So the information present in the genome is able to code for the whole body itself, the structure of the cell and the structure of the organism as well. And this happens because there are several degrees of informational transactions. And that is where we come to something, the central to all of these things comes to the central dogma of life or molecular biology. The DNA, the information that is present as DNA, maybe as the genes. Genes are a specific sequence of nucleotides which would encode for a protein or an RNA. So the DNA, the information that is present in the DNA is decoded by RNA polymerase, an enzyme which is DNA dependent RNA polymerase, which through the process called as transcription will generate mRNA or just RNA. If it is mRNA, the mRNA would be recognized by, that is messenger RNA, by ribosomes, which are large ribonucleoproteins, and they would translate this, the mRNA, the sequence, the ribosome would read the bases on this and make specific sequence of amino acids, polymerized specific sequence of amino acids, and that is called as a protein. The gene would give rise to a protein, and it will always be the specific sequence. So the sequence of information from DNA, it is first transcribed to RNA. Because it is still in the nucleotide language of nucleotides, here to here, we call that as transcription. It is like something like rewriting what is in capital letters in small letters, for example. And from the sequence of RNA, we are generating protein. That process is called as translation. And because the sequence is read from the language of nucleotides and a sequence of amino acid is made, we call that as translation. Or you can think of it as from the language of nucleotides, now it is in the language of amino acids. Therefore, we call it as translation. Ribosome is a RNA dependent amino acid polymerase. You can also think of it that way. There is one important thing that we have to notice is DNA replication. This is the most important step or quality of every genetic material. You can take any gene or any fragment of DNA. It should be having the capacity for replication. If you take suppose you take a tube or a bottle which has a DNA in it, would you call that genetic material? It is not genetic material. It is just a chemical. It would become a genetic material only when it is in association with an origin of replication. Origin of replication is where initiation of replication would start. And the process is called as DNA replication, and the enzyme name is called as DNA polymerase. So I hope you will be able to remember this, that any DNA that has the origin of replication or can be replicated or is in association with the replicator, then you call it it is genetic information. It is genetic material. If we cut out a gene from a genome and put it outside, it is chemical. If you take a gene from outside and put it in association with a replicator, then it is a genetic information, genetic material. It is just a philosophical way of how to see it. The context is important. And out of all these processes, DNA replication is the most essential one. Say, for example, plasmids have the property for DNA replication. They may not necessarily, or viroids, for example, they have this RNA replication. So replication is the primary aspect of any genetic material. So if you see, there are some more, there is reverse flow of information as well. That is, from RNA, we can there are systems where DNA is synthesized, and that is called as reverse transcription, because you're writing from the language of RNA to the language of DNA. Both are still nucleic acids, and it is the reverse of transcription, so we call it as reverse transcription. And there are also several others, especially we would find these in case of viruses, where you might have a plus strand and a minus strand, which we will discuss later in the later classes. But otherwise, we have to remember this. What is the informational wise, what is the reactant, and what is the enzyme, and what is the product. Say if I want to describe DNA polymerase, DNA, this is all with regard to information, okay? So here for DNA polymerase, for example, information is DNA, and the product is, it is DNA. So that is, the enzyme is DNA polymerase. We would then say it as DNA dependent DNA polymerase. Similarly, we can also,